Researchers have designed the first living medicine to treat lung infections. The treatment targets Pseudomonas aeruginosa, a type of bacteria which is naturally resistant to many types of antibiotics and is a common source of infections in hospitals. The treatment involves using a modified version of the bacterium Mycoplasma pneumoniae, removing its ability to cause disease and repurposing it to attack P. aeruginosa instead. The modified bacterium is used in combination with low doses of antibiotics that would otherwise not work on their own. Researchers tested the efficacy of the treatment in mice, finding that it significantly reduced lung infections. The living medicine doubled mouse survival rate compared to not using any treatment. Administering a single high dose of the treatment showed no signs of toxicity in the lungs. Once the treatment had finished its course, the innate immune system cleared the modified bacteria in a period of 4 days. The findings are published in the journal Nature Biotechnology. P. aeruginosa infections are difficult to treat because the bacteria lives in communities that form biofilms. Biofilms can attach themselves to various surfaces in the body, forming impenetrable structures that escape the reach of antibiotics. P. aeruginosa biofilms can grow on the surface of endotracheal tubes used by critically ill patients who require mechanical ventilators to breathe. This causes ventilator-associated pneumonia (VAP), a condition which affects 1 in 4 patients who require intubation. The incidence exceeds 50% for patients intubated because of severe COVID-19. VAP can extend the duration in intensive care unit for up to 13 days and kills up to 1 in 8 patients. The authors of the study engineered M. pneumoniae to dissolve biofilms by equipping it with the ability to produce various molecules including pyosins, toxins naturally produced by bacteria to kill or inhibit the growth Pseudomonas bacterial strains. To test its efficacy, they collected P. aeruginosa biofilms from the endotracheal tubes of patients in intensive care units. They found the treatment penetrated the barrier and successfully dissolved the biofilms. The researcher says that they have developed a battering ram that lays siege to antibiotic-resistant bacteria. The treatment punches holes in their cell walls, providing crucial entry points for antibiotics to invade and clear infections at their source. The researchers believe this is a promising new strategy to address the leading cause of mortality in hospitals. With the aim of using the living medicine to treat VAP, the researchers will carry out further tests before reaching the clinical trial phase. The treatment is expected to be administered using a nebulizer, a device that turns liquid medicine into a mist, which is then inhaled through a mouthpiece or a mask. One of the advantages of using M. pneumoniae to treat respiratory diseases is that it is naturally adapted to lung tissue. After administering the modified bacterium, it travels straight to the source of a respiratory infection where it sets up shop like a temporary factory and produces a variety of therapeutic molecules. By showing that M. pneumoniae can tackle infections in the lung, the study opens the door for researchers creating new strains of the bacteria to tackle other types of respiratory diseases such as lung cancer or asthma. The bacterium can be modified with a variety of different payloads, whether these are cytokines, nanobodies, or defensins. The aim is to diversify the modified bacterium's arsenal and unlock its full potential in treating a variety of complex diseases.